Students, in continuation to the previous lecture, we will be dealing with the factors which influence the spermatogenesis, or which control spermatogenesis. The factors include the hormonal, the environmental factors, the hormonal factors include the hormone which is FSH secreted by anterior pituitary. The environmental factors, they include light and temperature. Other factors besides hormonal and environmental factor include the psychological state of mind, of the individual, the physiological state of health, of individual, and the age of individual. That's it. Now, today we will be taking up oogenesis, which is the gametogenesis involved in the formation of ovum. Simply, oogenesis is a process where the ovum are produced in ovaries, and oogenesis is almost a similar process as was seen in case of the spermatogenesis. But oogenesis takes place where? In the female individual. And as we have already discussed, the organs where the oogenesis takes place in female are ovaries. In all vertebrates, we have got the paired ovaries except for the birds. So birds have just one ovary, as another has been lost for the want of reduction of the weight, which is an adaptation to the flight. So one thing I would like to tell here is that the oogenesis is a process which is completed in 3 stages, and these 3 stages are similar to those found in process of spermatogenesis. So 3 stages found in oogenesis are again the same: multiplication phase, growth phase, and maturation. maturation phase. Now, before coming directly to the phases of the oogenesis, I would like to tell you, I would like to explain the structure of ovary, because what happens is that the whole process takes place in ovary. In different stages they are seen where in the ovary. So if we look at the LS of the ovary of, say, the mammals, it looks like an irregular structure, and the ovary, if we look at the histology of the ovary, in the center we have the central portion, we have got what? We have got the medulla. This is what? This is medulla. Medulla is the innermost part which contains what? The relatively large blood and lymph vessels. So what it contains is large blood and lymph vessels. Just surrounding the medulla, we have got what? We have got the cortex. Surrounding what? Surrounding medulla, we have got the cortex. Surrounding medulla, what we have got? We have got the cortex. And this area is represented by what? This area is represented by cortex. This lies outside the medulla, and it contains early or young and ripe developing ova. It contains what? This is cortex, which contains developing follicles, that is ova. Done. And then the outer portion, or we can say the outside the cortex, is the area which is called as tunica albuginea. Tunica albuginea. So tunica albuginea, it is a connective tissue layer which surrounds the cortex. This is the connective tissue layer. Tunica albuginea is connective tissue layer which surround the cortex. So this is what? This is the LS of ovary. LS of ovary. Now one thing, the outermost epithelial lining. This is the outermost epithelial lining, and this is copy to whole the area. This epithelial lining is the germinal epithelium. These are germ cells. These are what? These are germ cell. That is the outermost covering or outermost epithelial lining include what? It constitutes, it is made up of what? It is made up of the germ cells. Now what happens? How? What happens? That one of the germ cell, it does what? It disassociates. One of the germ cell, it disassociates from the epithelial lining, epithelial layer, and it starts proliferating. It starts what? It starts proliferating. That means it starts dividing. It starts dividing. It starts dividing. In the stage of, say, 8-16 cells stage, that is a bunch of cells is formed as the result of the division of single germ cell which had disassociated from the layer, and this bunch of cells is called as egg nest. This is called as egg nest. Now one of the cell of the egg nest which is destined to form the ova. What I'm saying? I'm saying that one of the cell of the egg nest which is destined to form egg, it increases in size. It grows in size, whereas rest of the cells of the egg nest, they surround it. The I would repeat once again that one of the cells of the egg nest which is destined to form ova occupies the central position, which is surrounded by the rest of the cells, and this, at this stage, we call it as the primary follicle. We call it as primary follicle. So this is what? This is the primary follicle. Now what happens? Now the primary follicle, it consists of what? The large cell which is destined to form the ova, surrounded by the follicular cells. These are the follicular cells. Now the follicular cells, they divide. They keep on dividing, and the number of layers of the follicular cells around the developing egg, they increase. So what I was saying is that the follicular cells, they keep on dividing and they form many layers. They form what? Many layers around the developing egg. Now what happens? I would just take your attention from this and will draw the diagram here of this, that this is the developing egg. This is the developing egg. So what I have drawn is the two layers around the developing egg. So the developing egg is where? That is in the center. So this is the first layer of follicular cells surrounding what? Surrounding the developing egg. And this is the second layer of cells. This is what? This is the second layer of cells around the developing egg. Now what you see, if I am able to make you understand through the diagram, that the cells of the inner layer, they are rounded, and the cells of the outer layer of the follicle are spindle shape. The inner layer is called as theca interna. This is called as theca interna. And the outer layer of the spindle cells is called as theca externa. Now what happens? Will be taking you back to the ovary, that these follicular cells, they keep on dividing, so the size of the follicle increases. And thereafter what happens is that the mature follicle is formed. And how it is formed? That when the number of follicular cells, they increase in size, they develop what? They develop a cavity. They develop a cavity in the follicular cells. So then I would just be drawing your attention to this diagram. This is the outermost layer of the cells which is called as theca externa, followed by theca interna, and then we have the developing what cavity, which is called as antrum, and into the antrum our hangs what the developing egg along with the cells, and these cells they are derived from what, another layer of cells which is derived from the theca interna, and that layer of cell is called as zona granulosa. That layer of cells is called as zona granulosa. So at the stage when the developing follicle develops what, the cavity, that is antrum, this stage is called as, at this stage the follicles they are called as Graafian follicle. So let us know the structure of Graafian follicle, because it will give you more knowledge about how the cell develops, how the ovum develops. The Graafian follicle, I would start drawing it from the outer side. That Graafian follicle consists of the outermost layer of the cells, which, as I already told you, are the spindle shape. This is theca externa. Theca externa consists of spindle shaped cells, followed by theca interna, so this is so theca externa is followed by the theca interna, and theca interna, as I already said, that theca interna consists of what? It consists of rounded cells. Whereas theca externa consists of what? The spindle shaped cells. So theca interna is followed by what? Theca interna is followed by zona granulosa. Theca interna is followed by zona granulosa, and again zona granulosa consists of what? Rounded cells which are derived from theca interna. But the cells of zona granulosa are more compact, or what? Are more compact with larger nuclei, So this is what? This is zona granulosa. This is what? This is zona granulosa. Again, so I would just like to correct myself that it is not zona granulosa, it is stratum granulosa. And the developing cell, it overhangs into what? It overhangs into the cavity. It overhangs into this is what? This is the cavity. This is the cavity which develops where? In the follicle. This is the cavity called as antrum. This is what? This is the cavity which is called as antrum. And antrum, or the cavity, is filled with semi-viscous fluid called as liquor follicle. This is filled with the semi-viscous fluid that is called as liquor follicle. And the developing ovum overhangs into the antrum, which is connected to what? Which is connected to stratum granulosa or zona granulosa through the narrow stalk like structure, which is called as the stalk, which is called as discus proligerus. The stalk is called as discus proligerus, or this discus proligerus is also called as cumulus oophorus. It is also called as cumulus oophorus. This is what? This is the stalk through which the developing embryo, sorry, not embryo, but the developing egg, it overhangs into what? The antrum, which remains filled with the liquor follicle. Thereafter, now we will be coming to what? We will be coming to the process of oogenesis, because whole of the structure of the follicle, that is Graafian follicle, is now very well understood by you. That is what I can hope. So what happens that when the Graafian follicle is formed, it ruptures. What happens? It ruptures. Why it ruptures? That you already know. For releasing the developing egg, or we can say now the egg is completely developed, ovum is completely developed, so it is released, and the leftover part of the follicle is called, it forms the corpus luteum. It forms what? It forms corpus luteum. So corpus luteum, the cells of the corpus luteum are called as luteal cells. This is corpus luteum, and the cells, they are luteal cells. And one thing for your knowledge is that the luteal cells, they are derived from what? They are derived from zona granulosa and theca interna. They are derived from what? Zona granulosa and theca interna. Another point, the corpus luteum, it forms what? It forms the endocrine part, as it secretes what? The progesterone, that is the pregnancy hormone. And if the fertilization not takes place, or the egg fails to fertilize, then the corpus luteum, it degenerates, and a simple white scar in the cortex of ovary is left, and this white scar, which is left as the result of degeneration of corpus luteum, is called as corpus albicans. Corpus albicans. And what happens to the corpus luteum when the egg gets fertilized? It continues secreting the progesterone till what? Till the end of pregnancy. So this was all about what? This was all about the structure of ovary, how the egg or ovum develops in the ovary. Now what actually the process of the development process of the oogenesis is that, again, the three phases, that is multiplication phase, growth phase, and the maturation phase. In multiplication phase, what happens is that the primordial germ cell, what the primordial germ cell, that is PGC, it starts proliferation. It starts what? Proliferation. Divides it divides and redivides. And when what? When the division starts, that is when the division or proliferation stops, the PGC, that is the primordial germ cell, is called as egg mother cell or oogonium Now what happens to the oogonium is that so this is the multiplication phase. This is the multiplication phase, and the oogonium, it undergoes what? It undergoes the growth phase. It undergoes growth phase, that is the oogonium undergoes growth, and it grows many folds. In spermatogenesis, what we found that the spermatogonium that underwent growth to form what? To form the primary spermatocyte, and the primary spermatocyte used to be double that of what? Used to be double that of the oogonium. That is, the size of primary spermatocyte was almost double the size that of the spermatogonium. Now, in this case, in oogenesis, the oogonium is far smaller than the primary oocyte. That is, the primary oocyte is manifold larger as compared to the oogonium. And one thing which I would like to mention here is that, similar to what we saw in spermatogenesis, the number of chromosome in the primordial germ cell is 2n. It is diploid, and the multiplication, and during multiplication, the division is mitotic in nature. So the oogonia have again what? The 2n, that is diploid number of chromosome, whereas in case of the primary oocyte, again we have the 2n number of the chromosome, that is, the primary oocyte are again what? The diploid in nature. What is worthwhile to mention here in growth phase, that the growth phase is very important. Growth phase is very important. Growth phase is of great importance as far as oogenesis is concerned. Why? Because, and growth phase is is of great importance and is subdivided into two phases. One is previtelogenesis, and other is Vitellogenesis. We will be taking up the pre-vitellogenesis, and during pre-vitellogenesis what happens that the cytoplasm, the nucleus, they observe large number of changes. They observe what? They observe large number of changes. The changes which are observed in cytoplasm and nucleus during pre-vitellogenesis, they include the growth of cytoplasmic substances. They include what? The growth of cytoplasmic substances. So in cytoplasm, various cytoplasmic substances, they grow in number, they grow in size, and some new structures they are developed when during the pre-vitellogenesis. So what are the changes exactly? They include what? The changes in number of mitochondria. The number of mitochondria increases during the pre-vitellogenesis. Mitochondria are fewer in young oocyte, but the number increase during the growth phase. The number increase during growth phase. Another point is the change in Golgi bodies. So the cytoplasm of young oocytes contains numerous vesicles surrounded by simple membranes. And what happens is that the Golgi bodies, they do what? They lead to the formation of cortical granules. And one thing I would like to tell you is that cortical granules are of great importance as far as they help in the formation of fertilization membrane. During fertilization. And this fertilization membrane does what? This fertilization membrane, as you might be knowing, it helps in preventing polyspermy. That means when one sperm enters the egg, another sperm cannot enter the same egg into which the sperm has entered, the first sperm has entered. So for that matter, the egg which undergoes fertilization, that develops the fertilization membrane, and the cortical granules which are derived from what? The Golgi complex, the Golgi bodies, they play a great role in that membrane formation. So when the cortical granules are formed, the Golgi bodies, they disappear. The Golgi bodies, they disappear. And then the endoplasmic reticulum, as far as endoplasmic reticulum is concerned, we find only what? We find only vesicles. With ribosomes. In the cytoplasm of what? In the cytoplasm of the oocyte. Thereafter comes Thereafter comes what? Thereafter comes the second phase, that is the vitellogenesis. And vitellogenesis is one of the very important phases. One of the very important phases which are seen during what? The oogenesis. During vitellogenesis, what happens that yolk is deposited, is synthesized and deposited in the oocyte. As yolk is the only source of the energy, acts as reserve food. So yolk acts as the reserve food. It helps, or it provides the energy during the development of the embryo. It provides the energy during development of embryo. Now, one thing, that in most of the organisms, in most of the vertebrates, yolk is formed where? In liver. Yolk is formed where? In liver. It is transported through blood to the developing follicles, and through the follicular cells it enters the developing oocyte. It enters. So in, as I said, that in most of the vertebrates, except for the fishes and amphibians, except for what? The fishes and amphibians, the yolk is synthesized where? Synthesized in liver. And then what happens to the yolk synthesized in liver? I already told you. And thereafter, and in fishes and amphibians, the yolk is synthesized where? In the modified mitochondria, which are present where? In the developing ovum itself, that is oocyte itself. Now, the yolk is a complex food component which consists of, or whose composition, if I would tell you, it contains what? The proteins, the phospholipids, and fats. So what we found, that during growth phase, which is very important phase of the development of the ovum, there are changes which take place in the cytoplasm. And one thing more which I could not discuss is that the changes which take place in the nucleus during the pre-vitellogenesis include the transcription, the synthesis of mRNA, the synthesis of tRNA, the synthesis of rRNA in the nucleoli, and besides this, the nuclear shape increases in quantity, and at this time the nucleus is also called as germinal vesicle. And thereafter we come to the maturation. We come to the maturation. In maturation, we would just be looking at this stage, that the primary oocyte, it undergoes maturation, and maturation is nothing but the meiosis. And meiosis, as you all know, has two steps, that is meiosis first, which is actually the reductional division. But what happens in the meiosis first is that the division is unequal. That is, the one daughter cell is of larger size and another one is smaller, and another one is of smaller size. And actually the meiosis first is the reductional division. So the number of chromosome in the smaller and larger one are what? The half. So they are haploid. The smaller one is called as the first polar body, and the larger one is called as secondary oocyte. Now, why the division is unequal? This is because the developing egg cannot afford to divide equally, because by dividing equally, what will happen is that the reserve food material and the cytoplasmic components, they will be shared equally, they will be divided equally. So there may face the shortage of reserve food material, that is the yolk, for the developing embryo. So what happens is that in this type of the division, only the chromosomes with little cytoplasm, they go to what? They go to the polar body, and maximum of the cytoplasm along with one set of the chromosome, it remains with the secondary oocyte. In this secondary oocyte, it undergoes the second phase of the meiosis second, and again the division is unequal, resulting into the formation of another polar body, and as we already know that meiosis second is similar to the equational division. So what happens is that we get is second polar body and ovum, and the first polar body, it divides, it undergoes the meiosis second to form two polar bodies. So in nutshell, what I would like to tell you here is that as the result of the oogenesis, as the result of oogenesis, we get only one ovum, we get only single ovum, and the three cells, the three polar bodies, all these three polar bodies, they get degenerated. They get what? Degenerated. All the three polar bodies, they get degenerated, and we get just one